Hi, I'm Dr. Sarah Redmond. I'm a professional biologist, but in my spare time, one of my hobbies is thinking about how organisms that are fictional might actually work in a real, biologically sound way. And so today I'm going to be talking about how undead organisms, zombies, might actually be encountered in the real world. So first, I'll acknowledge that there are some cases that are real, that have a lot of similarities to what we consider in the fictional portrayals of zombies. And a lot of these have to do with toxins that are able to kind of hijack brain activity or limit brain activity in a way that impedes decision making or removes a lot of the characteristics we would associate with personality in humans. So one of these is the tetrodotoxin family. These are found in pufferfish and some other prey animals. They are common in the voodoo rituals in Haiti, which is one of the main places where we had a lot of early stories about zombies, and there have been a few recorded cases of people who were exposed to these through rituals that caused them to lose a lot of their normal human characteristics and become zombie-like. There are also biological organisms, some bacteria, some Vibrio species, that can produce similar toxins at a low level. So it would have to be something like a mutant strain or an engineered strain to produce a high enough level of toxin to be really effective as a transmissible zombie pathogen. And then there are also some parasites that can inject toxins into their hosts and use that as a way of controlling them. So I've given here a picture of the jewel wasp that you can see here. It's injecting into the brain of a cockroach, and what it will actually do is once the cockroach is immobilized, it will pull the cockroach, and the cockroach will still walk with it, but it doesn't move on its own. So it will pull the cockroach to a safe location, lay its eggs inside the thorax of the cockroach while the cockroach is alive, and then when those eggs hatch, the larva of the wasp will eat the cockroach from the inside, and that's the point when it actually dies. There are other parasites that are both larger, some insects and some fungi, as well as some microbes that can control brain activity. There are even some microbes that can cause their host to have desires to seek out certain environments that allow the microbes to survive more successfully. Okay, so brain activity is a big part of what we consider to be a zombification event. If you lose your brain activity, then even in colloquial terms, we sometimes refer to you as a zombie, like a TV zombie. And so looking at healthy humans versus undead humans, we would expect to see that there's going to be some difference in brain activity. So I've pulled some scans just to kind of give examples of what we might expect. The key thing is that your cortex here is going to be a lot of what we consider higher brain function that's associated with decision making and making who you are. Whereas the brain stem is associated with more basic survival, so things like breathing, not a whole lot of temperature regulation, but a little bit. Just basic processes are associated more with this brain stem area. And so we would expect that in healthy humans, we have a lot more activity in this upper area of the brain. And you can recall, if you think back to a lot of fictional portrayals of zombies, it's not just destroying the brain, but that lower part of the brain right at the base of the spine that's important. Now, interestingly, I actually pulled these images from a paper that was published looking at how cough and cough suppression looks in your brain. So these people were put into the MRI, and they're all asleep, and the ones on the left are being induced to cough, and the one on the right is not. So you can see that coughing actually requires activity of these higher brain areas. So it's possible that zombies might not be able to cough in response to something introduced in their respiratory passages. In this case, it was capsaicin from hot peppers. So if you are thinking someone might be a zombie, shove some hot pepper in their face and see what happens. So if you're in a zombie outbreak, how can you actually use biology to your advantage in order to survive? I've kind of set this up as a 4-step semi-scientific process. Obviously, we're not going to reach the ideal end of science in reporting our findings to a broader scientific audience, because our key goal here is survival. So, the first step is going to be observing, and observing has 2 key parts. One of them is actually watching, and for the watching, it'd be nice to have some tools like binoculars or a telescope, but also a secure location with a good range of visible area around you, so that you can hopefully see a wide range of zombie interactions and normal zombie behavior. But the second really critical part is recording your observations. So keeping detailed notes of what you see, perhaps time of day, temperature if you can measure it, weather conditions, numbers of individuals that you're observing. Those are all going to be important for understanding both how individual zombies work, so whether they're attracted to the sound of an ice cream truck, or whether they're able to climb a fence, but also understanding how the outbreak might be working. When you see 5 zombies one day and 50 the next day, that tells you that it might actually be a pretty rapid rate of spread. So keeping notes on a lot of detailed information will be important for how you respond to this situation. Okay, one key thing that you would want to try and identify is how the zombification process actually spreads. This is partially to protect yourself because you don't want to become one of the undead, but also to help you think about how you can best combat it if you reach the point of looking for a preventative measure or a cure. So some common mechanisms we might think about: direct contact, whether this is like a short-range respiratory transmission, so if someone coughs on you, do you become undead? If someone bites you, if someone gets their blood on you. These are all common mechanisms for transmitting diseases that might be relevant, especially if we're dealing with an infectious agent. If we're dealing with an environmental toxin, is there a certain location where people go and then become undead? Is there some chemical that they might be coming in contact with, or some surface, something new or unusual in that location in the environment? And then another option that we see sometimes in fictional portrayals of zombies is what I'm calling corpse conversion, where individuals die of normal causes, they are buried or put in a morgue, and then they reanimate as undead individuals. These all have different significances about what it means for you as someone trying to survive the outbreak, and for someone who's trying to treat the outbreak. Additionally, it would be useful to keep notes on which species are affected. So I've kind of ranked these in order of biological similarity to humans. So if it's only humans being affected, which in fiction it often is, then that gives us information that it's a toxin or infection that is affecting some protein or other molecule unique to human biology. If we see that primates are affected, number 1, you have to worry about zombie chimpanzees, and number 2, that tells us something about this particular group of organisms is unique to the way that this particular undead process is working. Similarly with all mammals or all vertebrates, there are not a whole lot of infections that affect these broader groups of organisms. The rabies virus is an example of a virus that has an impact across a broad range of species. It's also a very, very lethal virus. And so if we see broad ranges of impacts, that suggests that there's something in the biology of all those organisms that can be hijacked by the zombification process. And there are other things that you might want to consider recording. I've encouraged you to think about other observations you might want to make, whether for your survival purposes or just to learn more about the outbreak. Okay, so once you've made observations, we're going to make a model for how the outbreak is working. This is an example of one that was published in a mathematics journal. So here we have humans dying naturally. Some of them can be reanimated. Sometimes zombies can die and be reanimated, so we have a bit of a cycle here. We also have healthy humans that might go through a zombification process. So in this case, there's actually 2 ways that you might become an undead individual. That's not a great case scenario because it means that it's going to be harder to identify people who are resistant to becoming zombies. And in this model, they were looking for whether it would be beneficial to treat. So you can see we've got the basic model with susceptible individuals in blue and the increasing zombie population in red. Initially, it looks like treatment is not doing a whole lot, but you can see there's 2 important things. Number 1, the point where these populations are equal is almost doubled. So we've got just under 3 weeks versus just under 6 weeks. And then also the blue line here ends up going to 0, but in the treated model, we do have a small population of humans that's completely surviving the outbreak. So if we reach this point where we're going to try and develop a treatment, what are some things to consider? We're going to have to investigate the zombies at some sort of close range. So we're going to need protection, and this means both protection from the undead and what they might do to damage you, but also from whatever's affecting them. So gloves, highly encouraged. Also a way of disinfecting the surfaces that you're working with and coming into contact with. 70% ethanol is a really common way of doing this in the lab. Bleach would be another good one. So if you can find a drugstore or a liquor store, you should be able to find sources of disinfectants that will be useful for this. Okay. Also, you're going to need a sample or maybe multiple samples of zombie tissue. Try and obtain those safely. And you're going to need some ways of making that tissue more easily visible. We have common lab stains that we use in research, so if you're near a university or research center, you might get those. But you might also be able to replicate some of these effects with things like food coloring or Kool-Aid if those aren't available to you. We're really just looking for differences in how the chemicals are attracted to different parts of the tissue. And then a microscope would be ideal. There are ways that you can fashion rudimentary microscopes that work well with a smartphone. So that's a hack that's not that hard to pull off with modern technology. Okay, so you're going to be comparing samples from your zombie to normal. For these purposes, you are normal. So this is what the outer skin layers look like in a normal, healthy human. You can see we've got some blood vessels. We've got nice, tight layers. There's not a lot of white space in this sample. And there's also some differences in how the stain is being bound to some of these different cell types. Here we've got an example of what that would look like in a necrotic tissue sample, which is something we might see in an undead individual. There's lots of air spaces. These red blood cells are clumped together. They're not they're not associated with the blood vessels. They're just kind of making a clot. We've got some white blood cells here. Those are the darker purple spots. These are all signs of a tissue that is dying. Okay, some other things to look for is whether the immune system is responding. So looking for those dark purple white blood cells. This is kind of a fuzzy picture, but a lot of them have unusually shaped nuclei, and that's a good way of identifying and characterizing them. If the immune system is responding, that suggests we might be dealing with an infection, and so you might lean more towards that rather than an environmental toxin. We could also look for foreign cell types. If we think it's an infection, we might see fungal cells that have a really thick cell wall, protozoa or bacteria. These are things that you could treat with an antimicrobial agent. Unfortunately, unless you have access to and can operate a scanning electron microscope, you will not see viruses or even just protein toxins affecting these individuals. Okay, so for treatment options, if we reach that point, a vaccine would be ideal to prevent new individuals from being converted into the undead, especially if you can identify individual humans or non-human species that are resistant to the zombification process. You can use the serum, the liquid components of their blood, as a way of preventing the spread in other individuals, their antibodies. If it does seem to be an infection, you might also consider antimicrobial drugs to kill the pathogen and prevent it from taking over an individual. In order to cure, we're probably looking at some way of stimulating the brain or cellular regeneration processes. So good luck.